So we had one last week, this week, yet again. And it's kind of nice how what we'll be talking about today is sort of an extension of what we talked about yesterday. But in a very specific, so we're taking that same concept as we talked about last week, and we're taking it into a wholly new direction. So what I'm going to talk about today is how our brains can work with information that's encoded in coordinates of the world around us. So what that means is that what we've been talking about up until now has been like a sensory frames of reference, if you will, but we also know how to navigate. And whenever we turn around, all of our sensory inputs change, right? But still we know where we are in the world. Our brains do have to know how to work with the world around us, and that is an abstraction. So today we'll be talking about how our brains create that abstraction. And I'll give it away now. We don't know. But we know that our brains contain this abstraction. And so I'm going to show you examples of how we know that this is the case, how these lines of evidence came about. And I hope you'll get an idea of why this was Nobel Prize winning territory. Yeah, well, let's go. So what we talked about, or at least what the intro was last time, was this idea that we have these receptive fields we can measure either in a neuron or in a sensor or a field of like a population of neurons or an fMRI voxel. Measure these receptive fields, and they exist usually in sensory coordinates. At least that's where the idea came from, right? From the 50s, the movie that I showed you, these receptive fields. Those are all sensory coordinates. And I did show you how you can take that metaphor further. But generally, the idea comes from this idea of sensory receptive fields. Yeah. And what that means is that you'll have this neuron or population of neurons fire for something, and that something has a selectivity specific to a location in what you might call the sensorium. I really like that word, so I put it in here. Right, it's the totality of our senses, right? So it's not just the retina, it's not just the cochlea, where sound comes in, it's not just the surface of your skin, but it's all these things together which form the interface between us and the world. Now, these receptive fields in the sensorium, they're very local on the surface of your skin, for example, or where you are on the retina. What we usually do is we describe these with these Gaussian models. Yeah, I talked about this model that we use to describe these population responses in terms of visual field coordinates, for example, and we use the Gaussian function for that, which is like the normal distribution. Has a very standard shape, and it's a very sort of statistically robust definition. If you have many different sources of noise, well, you'll end up with something that is shaped like a normal distribution. So it's like a sort of standard statistical sort of thing. It turns out that, for example, in terms of vision, we can describe these population responses as a Gaussian blob somewhere in visual space. We'll talk about that last time, right? But this you can sort of take to any sensory dimension. We also saw this. So what I'm just showing you here is how one of these units, right, be they a voxel or a neuron, would respond. So this could be firing rate or hold response or whatever, along some sensory dimension. Yeah, I'm just plotting it there. This is the way that we tile this space of information, this sort of dimension of information. So in this case, I'm putting it here like tone frequency. You see that they are becoming wider as you go to the right. You get this sort of sampling that you can look at in this space. So you can really get a handle of how the brain represents this sensory modality, right? You can also have, for example, visual eccentricity. Turns out that, I don't know if you distilled it from the last lecture, but eccentricity, so there's the distance from where you are looking, right, towards more eccentric regions, the periphery. We call that visual eccentricity, and tone frequency. They are actually very, very similar in terms of that they are sort of log spaced. It's a very beautiful sort of relevant thing. But you can also do this in terms of position on the body map. We can extend this into abstract dimensions. Like you also saw, right, like semantic spaces or numerosity, and so forth and so forth. I'm so glad that everybody's sort of a bit late today. So thanks for that. Yeah. So we discussed this a bit the previous lecture. Like we can actually look at how the sensorium in this specific dimension is tiled, and we can say things about what this means for function. Right? For example, in the case of numerosity, there is a slight overrepresentation of the smaller numbers. Well, those do occur more in everyday life, right? So it makes sense to have a more stable, a more overrepresented representation in the brain for those. Yeah? And the same goes for like everything close to where you are looking. That the fact that we can move our eyes means that if we have a very dense sampling around where we are looking, that is really beneficial for visual function. So you get these maps, right, where you get a correlation between the preferred numerosity. So this is a plot where this number, the color then, stands for the location of this peak, yeah. Whereas this is tuning width, so that's a different quantity. That is the width of these Gaussians. Yeah, so that's the standard deviation, if you will, of the Gaussian function. And so what we're looking at here is a map of preferred numerosity and a map of preferred tuning width. You see that they are highly correlated, right? This means that you have smaller receptive fields for those quantities that are also smaller. And it means that you're better at discriminating smaller quantities also. This is a sort of standard sampling a thing that we that sort of pass by. Yeah, the standard thing that we take from sensory, the same is true for vision, for example. The receptive fields near your fovea, where you are looking, are very small, and there are a lot of them. Right? So you're just very good at that because of this. So close to the fovea in this case also means smaller receptive field. I showed you some plots of that in the previous lecture. Oh, here we go. Yeah. So high preferred numerosity and wide tuning are correlated. And that's the nice thing about this. Also, you can just plot it on the surface of the brain and you see it. Right? Oh, there's correlation there. Right? You have to sort of invert the color scale, and they use the same color scale, which wouldn't be my preferred option. But you get these things are correlated, and it tells us something about how this sensory sampling works, how the brain represents these sensory dimensions. There are specific rules to this. But if you have these receptive fields in sensory coordinates, I see all of you looking at this right thing. What the hell is that? I'll show you in a bit. So these receptive fields in sensory coordinates, they constitute a problem. I already told you. It's like, well, you don't have this problem so much, but I do right now. Right? It's that all of these inputs that come in through my senses, definitely, they're changing all the time. And what that means is if you want your senses to tell you something about the outside world, and for now, let's assume that the outside world is stable, yeah, you're going to have these changing impressions all the time. How do you make sense of that? How do you relate that to what's going on in the world? Yeah. So it really is the job of our brains, based on sensory information, to distill what the world is like, right? Because we need to act in the world. We need to find food, shelter, water, a mate during our lifetimes in the real world. Right? And so we need to have this function in the real world. That's where the litmus test is of our sort of evolutionary selection. You want to go really broad. So it's nice to understand these sensory coordinates, and it's nice to understand how information is processed in these sensory coordinates, because that's the vat our brains live in, right? There's no getting around these senses. But we do understand, of course, that the brain needs to transform all of these changing sensory impressions to something that we can relate to the actual world, right? If we're going to have a break and I want to walk to the coffee machine, I need to like plan in a very abstract way, right? It's totally divorced from what I'm experiencing right now, what's coming in through my senses. And somehow that has been built up from all of my previous impressions that were sensory. Now, I just want to try to impress you, not myself, but with the Herculean task that the brain is solving when it is able to perform this sort of thing. Right? To be able to abstract the world coordinates from these changing sensory impressions, if you think of what that means in terms of what the task is that the brain is solving at that point, it's really, really, really difficult. So here, the example here is what happens in terms of vision when you move your eyes around. And so what you see here is an image where this represents where an observer is looking, and the observer is just going to look around the image. Now, what that means is that this central portion of your visual field, where you're really, really good at seeing, right, is going to be jotting all over the image. And outside of that very high resolution part of our retina, we don't really see that well. And that's that's sort of communicated by this sort of black here. So try to imagine that you're not seeing the thing on the right. I've already shown you, but still. And just look at here and imagine, how do you distill what's there? Right? So you see a couple of things, like it's changing very rapidly, right? And if you're basing yourself just on visual information, there's really no way of telling what came when, or like what is where, right? You just see these very disjoint sort of snapshots. And you see that it's shaking all over the place. Our eyes just shake all the time. And so this is the problem that your brain is having solved. Now, of course, this is sort of an exaggeration in one important way. Can anyone think of why this is an exaggeration? Like what does your brain do? Yeah, go ahead. Yes. So this is yes, you're right. This is a bit bogus. You do have peripheral vision, so you do know what is around there. So I'm exaggerating how little you see outside. But if you actually see, so I fully agree with you, and I'm exaggerating here. At the same time, this problem is way worse than you thought. The part of your vision where you actually see color, for example, is only like this big, right? Where you actually are able to distinguish colors. All of the rest is just sort of filled in from the center outward, and by your ability to make these eye movements all the time. The other thing is our retinas are really badly architected. So like God was asleep or something, because we have all of the vasculature that provides oxygen and nutrients to the retina. It's actually on top of the retina. So there are huge like webs in your field of vision where there's like these blood vessels running over it. You can't see there, but you don't know that. Your brain just fills in.
You even sometimes see the blood actually.
Oh yeah. So there are. Oh yeah. So those are like little phosphines. It's like the sort of noise in the photoreceptors that you can see. The noise, they can also be like retinal ganglion cells. The retina is a beautiful, like very complex machine already. So that you sort of have these. Yeah, if you put pressure on your eye, you can start to see these things. Yeah, it's quite interesting. Yeah, I was actually I was meaning to talk about this other thing that your brain does do, and so I'm gonna go ahead.
But also we keep track of our own eye movements.
Exactly. Yes. So so one of the exaggerations was that I'm making you blind in the periphery or not. Fair point. The other is you're not just experiencing this passively, right? You are actually actively sampling the visual world around you, and you are actively foraging for visual information. And this loop is extremely important. And I won't go into it specifically here, or like in this lecture, but it is a very, very important component that in many cases, people in neuroscience have not thought about enough. And what I mean by that is, you put people in an MEG scanner or an fMRI scanner or whatever, or, you can try to do electrophysiology on a cat or a monkey or a mouse. Usually what you need to do is make them hold still, right? And so eye movements are a bit of not an example of that sort of thing, but like actively foraging for things is really hard to research, because people and animals will move. Even though it's important for a function, that makes an experiment really hard to perform well. Okay, I showed you this a bit, right? We have the retina on the bottom and the hippocampus at the top. And I already told you, like, there's a gradient of extremely beautiful abstraction going on from the bottom to the top. And we talked about this in that previous lecture in terms of vision. You see this happening. I want to highlight one specific aspect in this lecture, and that is what happens right at the top here. So you see these two regions, ER and hippocampus there, and that's where we'll be focusing on now. The specific types of coding that happen at the top levels of this hierarchy. Because in order to get those world-centered representations that are so important for our everyday life, our brain needs to integrate across different sensory modalities over time, and so forth and so forth. So all of those sort of gradients that I tried to highlight last time, like increased integration across the senses, increased time windows, all of those, they actually help our brains be able to abstract towards this level of world-centered representations, is the idea. But we know, for example, that action is important in this process of going from there to there. But in general, if you ask neuroscientists what is like the biggest question that we really don't know how to solve, people will give their sort of niche answers. But this question of how does the brain go from this low-level information to this high-level information processing, that really is a very, very open, partly because of these technical problems that I just highlighted, also the movement part. And so really is one of the biggest questions in contemporary neuroscience. So we do know, so I just told you, we know the beginnings, right? And we know this receptive field analogy really helps us understand stuff that happens from here to there. And we also know, just from everyday life, that we do use high-level world-centered representations, right? And so what do I mean specifically by world-centered coordinates? How do we actually have scientific evidence, apart from this introspection, that this sort of thing actually exists? And so I want to take you back to ancient psychology, when psychology and neuroscience were still very much related. So this is Tolman that you see there. So if we go back to the 40s, it's when the behaviorists ruled psychology. B.F. Skinner, for example, was a very large figure in that field. And they actually were very dismissive of the idea that animals and humans have like a very rich cognition apart from just input-output relationships. So their stance was really to try to explain behavior as a simple as possible combination of input and output relations. Trying to be very, very low level. And so what Tolman did was he had rats in this maze, and he taught them how to go from the start through the maze, quite a complicated maze this, to the food box. So they would learn where the food box was by following this path, and then a behaviorist would say, right, if you want to only talk about low-level stuff, the behaviorist would say, okay, now the rat must have learned to take a right, take a left, take a left, take a right, and so forth and so forth, right? Low-level information. The rat doesn't have a model of what it's doing. It's just doing simple input-output relation things. But what Tolman found was that if you were to, so he was smart, and his maze, he could just like put all these doors, take them out and put them back in, so he could change the layout of the maze, although the maze itself as a whole wasn't changed in terms of its in terms of the map of the maze. So he first learned that even without any reward, if you just let the rat just forage around in this maze and then place food there, the rat would be extremely fast in learning this. Really show that it had already formed a mental model of what that maze was like. So that's the first thing he showed. The second is, he showed that if he changed certain aspects of the maze, like I said, like change a little door there, here and there, it's not the case that the rat would need to relearn everything because, you know, at the third turn, I have to now take a right instead of the left. But the rat would be able to immediately understand where he or she would need to go in order to get to the food box. So, two very important indications that even without reward, the reward was also very important for the behaviorist as a sort of motivating factor for the organism, right? So even without having the reward there, the rat would already create a mental model of where to go this maze, so already know the world layout of the maze, and would be able to, you know, ad hoc find new routes based on this mental model. Yes. And so what that showed already in 48, behaviorally, is that you cannot explain this sort of thing from the behaviorist stance. Right? You have to, you know, account for the fact that apparently rats do have a mental model of what the world around them is like, right? What the layout of the world around them is like. Now he called this a cognitive map. And so it's not a sensory map, right? But it's a cognitive map. In many parts of this field, you definitely say a cognition high level, right? Now, okay, if we know that this is the case, we can take it one step further again, like we have now a notion of what exists, right, in terms of behavior. Can we find the neural correlate of this type of function? We've gone from introspection to now having a clear line of evidence that suggests that this type of processing must exist. And so people started looking for these types of things. And of course, people needed to develop a lot of electrophysiological skills and stuff. So only 20, 30 years later, people like O'Keefe discovered that if you go to hippocampus, right, that thing all the way at the bottom, at the top of that hierarchy that I've been showing you, if you record from electrodes there, they have very specific firing patterns that at first people really didn't understand what they were about. There was very sort of punctate firing. So you'd be near a cell, you'd record from its activation, and you would have this animal just forage around a bit through a cage, and you would find that it's sometimes very active and sometimes not, the cell, but they would not know why. Why here, for example, or why now. And it turns out that they found that if you just look at the top of if you take like the world view of where the rat is, and you plot its own position, so that is the position of the rat itself, foraging around an enclosure, and you then take the red dots are the spikes that the neuron fires. What you see here is that you have a lot of firing at this position and none whatsoever, and only a few spikes here and there around the enclosure. So what this means is, this is not the cell's receptive field. You can sort of think of it as a receptive field, but what they did was they called this the place field of this cell, because it reports not a location in sensory coordinates, but it reports a location in outside world space. So this is evidence of world-centered coding in the hippocampus. Now, O'Keefe was one of the people that got the Nobel Prize, but there's more. So one thing so I'll get into the thing that's more a couple of slides in later on. So one thing that's really interesting is if you think like, okay, well, we have like thousands and thousands, millions of neurons in the hippocampus, right? There's a lot of neurons there. So if you have these place cells and they're all over and they have different locations, right? If they tile the 3D world around us in the same way that these receptive fields in the sensorium I'm just gonna keep using that term, right? Are tiling that space, then you should be able to take a population of these cells, and then if you record from all of them, you should be able to deduce where the animal is in the space. Yeah. So from a population of these neurons, if the rest of the brain is able to listen to hippocampus, right, it should be able to know where it is in space. Right? And that means that as an animal, you know whether you should start eating in a specific location, or if you're in a completely different situation and you should be in a different location, you should be doing something else. So a beautiful example, right? You really see this beautiful high firing rate region in the world where that rat is right now. Beautiful finding, and this is half of a Nobel Prize. So if you're thinking about going into research, you need to do something like this, and that only gets you half a Nobel Prize. Yeah, yeah, it's just not. Yeah. Anyway, so you can then not just know where you are if you listen to a whole population. It also means that you have access to trajectories. So you can have one cell firing and another not. You know that you're in a specific location. But if you add time to the mix, you can actually have routes or trajectories by the temporal pattern of firing of these cells. Yeah. So it turns out that subsequent research was able to beautifully show that you know, hippocampus actually represents paths, so not just locations, but actual paths, which it then encodes as a sequence of activations of these place. But what's even more beautiful, right? Because this is sort of trivial, right? If you have a place field, then that's what you get, just activation. The beautiful thing is that what people also discovered, first during sleep, but also later during other moments, also during wakefulness, as people call it. So if you train a rat by letting it run through along this maze, and you have these 5 play cells, A, B, C, D, E, that fire in sequence. Yeah. So if the rat is running through that, it will experience first A, then B, then C, and so forth. And these specific play cells, they will fire at these moments in time. If this takes like 4 seconds, that's easy. That's just going from A to B to C to D to E, and going through the activations of each of these play cells in sequence. But it turns out that the hippocampus is able to do much, much more. It can do, for example, during sleep. This was found by, I think, colleague Bujaki. I think he's one of the podcast interviewees. And so what they found first is that if you record from hippocampus, there are these specific event triggers that will trigger what's called replay, so that this sequence of firing is actually replayed and much, much faster. So you have exactly the same temporal order, exactly the same route or trajectory through neural space that's being followed, but time compressed. Okay? And what they also found, and this is, I'm just here summarizing one slide, something like 15 years of electrophysiological research, right? So what they also found is this reverse replay, where you actually find exactly the opposite sequence of firing, right? So which is the same sequence, but reverse order. Like, how did I, if I were to go back really quickly, how would that work? So what the hell? Yeah.
They also look at when you place rat in the middle of the class. So for example, if you take rat out right now, if that still fires for C, or if that's because it's not found anymore.
Yeah, it's just an element, right? At that point. Yeah. So if you were to place a, so I'll get into this, but because you're having a very interesting idea, and people have had the same idea. This is what happens when you get interested in science. You start reading the literature, and then you have this idea of, whoa, we should do this. This would be such an awesome experiment. I had this during my PhD like 500 times. Very, very fatiguing. Because what happens is you go, it's an awesome idea. People should have, like, and then you go read it, and then they did it in 1967. They did it in 1972. Did it, and the thing is, as you grow up in science, this boundary becomes closer and closer to present, right? So I think I'm now 5 years ago, still not at the present day. So you just try to keep up. Anyway,
Is the replay connected, or the time of the replay also connected to the actual length of the sequence? Like, is it taking a bit longer at least when the path is longer?
Yes, yes, so that's the idea. The general idea is that what your brain is really, what the hippocampus is able to do is take something of this regularity of the sequence, and time compress it. But, yeah, it is the actual sequence, or at least the brain's reflection of the actual sequence, right? Because if somehow, I don't know, I'm just making this up, right? But if the rat really got distracted around D, then maybe that would just fall out of the sequence that is able to replay. But yeah. Yeah.
Going the sleep is it always move forward and backward?
Yeah. Yes, they, well, so, I think there have been a lot of, recent developments in this field, and it's not my field, so I can't really go into what the, like, really state of the art of the field is. But yes, when they found these two things, they were found separately. They sort of occurred at random. So your hippocampus is also very, like, it's not part of the model part of this lecture, but the hippocampus is really, really interesting in terms of just the electrophysiological properties of its function. So what that means is that you have these sort of hippocampal, what they call ripples, which is like a really fast sort of flicker of activation throughout, and that serves as a sort of reset that sort of triggers this type of replay events, but also triggers like whole cortex-wide association at the same time. And then these sorts of things are communicated via theta wave oscillations, and it's like a really beautiful electrophysiological machine. I don't know where people are in distinguishing whether some of these aspects are more associated with forward or reverse replay. First, they found forward replay. It's easier to find because it's the same sequence, right? Then they found reverse replay, and it gets weirder. So, right, like I would be able to tell you which specific information, which specific sort of triggering events lead to reverse or forward replay. So I don't know that specifically, but it's pretty out there that your brain is able to sort of replay in opposite order its experiences that it had hours ago. And you immediately also recognize, and I haven't focused on this too much now, that, you know, hippocampus is extremely important in memory. Right? But that's all sideshow. I just want to go, yeah.
Is it the same, similar example when you use warm-ups to bring up place and then you will use it to do the waveback, but you are able to do the waveback without, without using it?
Exactly, yeah. You would, as you are doing that, right? Let's say you walk from here to the station, Zuid station, right? Or you came from Zuid station, you'd be able to like retrace your steps in your mind. That would be reverse replay. Yeah, so that is like cognitively triggered reverse replay. And the reason that I'm saying that is it is because of what I'll show you in the next 1 or 2 slides. Because you know, after you found replay, the replay was found somewhere in the 80s, 90s. You start to think like, okay, but you know, this is awesome, right? This has to relate to like the contents of your cognitive of your cognition, and so how does this work? And I'm going to show you a couple of experiments that I think are really sort of exemplary of what this type of function does in the brain, right? What the function really is in terms of cognition. So this replay you can also see as a preplay. That is, it plays a role in planning. So the main idea is that this replay helps memory formation. So there, the idea is that you have this replay. It's you know, if you, let's say you have a sequence of events that happened over the course of 5 minutes, what your hippocampus is able to do is replay it really quickly, like within a couple of seconds. And what happens is the hippocampus is really nicely related or connected to the rest of the brain. So what that means is that the rest of the brain can sort of re-experience this thing that happened over the course of 10 minutes, but now in a very, very short time frame. And so it will learn associations between these different elements of the replay sequence. And that's the idea, is that hippocampus just replays these things, and then because it time compresses, the cortex can start to sort of absorb these connections. So the hippocampus serves to connect different more distance events in time. Anyway, so that's the reason why we think replay is helpful for memory formation. So if you look at this, you have this oscillatory coupling between the hippocampus and the cortex. So the cortex, cerebral cortex, the large part of the brain at the top, right? If you see more of this coupling during these ripples, that predicts how well you're learning as an individual, right? On a sleep by sleep basis. But it also, and so now we're stepping outside of the sleep part of things. This also supports wakeful behaviors, this sort of replay. So if you record from a play cell and it's supposed to go in some direction, if you see replay in the correct order, that would be correct for future behavior, then you see that the task is performed correctly. Whereas if you see replay of some other random stuff that is also in the maze, but isn't actually required for correct behavior, you see that you see this enormous difference in behavior, right? So if you get a replay that's associated with the actual task, that's correct for the task that the animal is supposed to perform, you get really good performance, right? As if the rat is sort of imagining beforehand, oh, now I need to do this and this and this and this. And by virtue of it having imagined this, it's able to perform the task. Whereas if you have some other replay where other play cells are firing for some random other stuff that's not relevant to the task, it will be near chance level.
Why the task relay is not one? Like why if you're
It's just stupid.
the same sequence? Why if you are able to do the same sequence, then it's not a guarantee that you're being.
Yeah. Exactly. So I think even if you were to do this in humans, you would not be at 100% because people get distracted and then they like at the last minute decide like, oh, I still want to go there or something like that. So there's noise in the behavior, definitely. There's also a criterion, of course, that you have to set as a researcher, like, okay, when do I call it task-related replay, right? What measure of similarity do I need to judge that this is the same? That it's, task-related replay. So there's a few of these things where, in general, what people do in like scientific practices, they do a t-test, like determining the difference between these two. And then when that's significant, they cheer. And so they're not that. What's important is that you relate these two and not necessarily this sort of ceiling. People don't care that much about that. And you can go even further. In a similar type of task, you can disrupt task relevant replay by just injecting electricity, injecting currents into the hippocampus. And what you get is that if you don't do anything, the rats are, the same level, more or less. They're able to perform the task, right? If you disrupt replay at a decision point where they're deciding where to go, you disrupt their ability to do it. Whereas if you do it at a location that's different, right? So this is the location where the rat is deciding where to go, right? That's when it has to have this mental model and start using it in order to decide whether to go left or right. That's when you impair its behavior. If you wait until after the rat has already made its decision and then start, fucking around with the hippocampus, no such effect, right? The animal has already used that replay related information to perform the task. So what these two things independently seem to point towards is that somehow this replay really seems to represent what the rat is thinking, right? If you want to use those words. So two reasons why we think that. The first is only when it relates to the task at hand do we see increased performance. And there's actually this causal manipulation that people have performed, you can actually, mess it up. And that's always a very important thing to be able to do. Let's have a break. How about that? Let's 7 minutes. How about that? 7 minutes, nice number. And we'll start again at 30.
En het subject zo weg waren.
So yeah. Now we have these place cells. We do. And they're pretty awesome, right? It's a very compelling finding. Still have a Nobel Prize on it, but it's very compelling. And so let's think about that. Let's say we have these place cells, right? How many places in your life have you encountered? You don't have to it's not, you can answer the question if you want. But, it's probably a lot. And so if you were to need a place cell for every specific location in every place that you've ever been in, you need a huge hippocampus, right? And so something is up there. Right? We found this beautiful concept, but we're missing something still. So how are we going to solve this problem, right? Because it does represent a problem. Yeah. So whenever we move to a different room, for example, our surroundings change, and our brains need to work with that. And how do place cells even keep track of these changes, like going from room to room, right? Are they it's a pretty cool sort of thing that happens. These neurons, they remap. So they have different place cell fields, so different place fields for any different set of surroundings there. So what that means is you put a rat in a circular maze of one, let's say color, environment one, that you find this place field for a specific cell. We're talking only about a specific cell. Then you place the rat in another environment, and it has this place field. Totally different. The shape of the surrounding is the same, but we change the surroundings and put different artifacts in the environment, and that will actually mean that it's a different environment to the rat. If you then go like a couple of days later, you place the rat back into environment number 2, you can actually record exactly the same place field from that same cell. Then place it like a week after back into environment number 1, and you would be measuring that place. So apparently, what the hippocampal cells are able to do is just go very easily from one environment to the other and have a completely different place field. Yeah? Now this is beautiful, right? Because you've just solved the problem of being able to encode space in many, many different surroundings. You've solved that problem. But what's the next problem? You already feel it, right? Yeah. Now you have how do you okay, now all of these hippocampal places have just shifted completely, right? How do you even know what's what anymore, right? In a new surrounding. So that's a really difficult problem yet again that we need to solve. Yeah. And I'll talk about one specific solution that people have come up with, like how does this remapping work? And how can it be sort of anchored still to the surroundings instead of just being like a random lookup table sort of thing on every new surrounding. So first, we want to understand like one of these recipes for how this remapping works. we want to go to, okay, what is this hippocampus? What does it look like? So hippocampus is what's called archicortex, so which is different from neocortex. Neo is new and archi is old. And what that means is it has a couple of layers. Neocortex has more, right? Newer things have more of whatever it is that, you know. So hippocampus has 3 layers. You can sort of see them here. It's sort of a layered, you can see the sort of layered structure. What you also see is that it has these sort of different sort of arc shapes to it. So this arc is called the cornu ammonis, which is like the arc of Ammon. I don't know why it's called that way. And then this one is called the dentate gyrus. So gyrus is also a word for gyrification or crown thing. And so this is like the standard sort of circuit of the hippocampus. And one thing that you're not seeing here is this that's not considered truly part of the hippocampus, which is just this part here, where you can draw a sort of circle, is this EC, which stands for entorhinal cortex. I don't know if you remember this from that diagram that I had with the hippocampus on the top and V1 at the bottom, but this entorhinal cortex was right underneath the hippocampus. And so the idea is that there is a very specific input-output circuit that the hippocampus represents. So the idea is that its inputs come from entorhinal cortex in this way. So here from entorhinal cortex, they are then projected into the dentate gyrus, and then that goes like this circle and then back out. So there's a specific input-output circuit that the hippocampus has. But you see that this entorhinal cortex is here on the left. It's sort of the gatekeeper to hippocampus. Right? So there has to be, if we are trying to understand hippocampus, we also need to try to understand what's happening in entorhinal cortex. This is the gatekeeper. If you don't understand what entorhinal cortex is doing, you are going to have a hard time understanding what hippocampus is doing. Turns out, entorhinal cortex is really fucking interesting. So we're hitting the jackpot once again, and this will allow us to move to the second half of our Nobel Prize. So this is really the reason that the place cells got their Nobel Prize is because of what was found subsequently in entorhinal cortex that sort of brought all of this together and really, has really started a really strong trend in neuroscience. So if you look at the conference that I was at, a lot of people are doing hippocampus entorhinal cortex type of work. It's like the burgeoning field, and a lot of that is due to the fact that only a couple of years ago there was a Nobel Prize to this field. People are like cows, right? Oh no, the Dutch saying is sheep. Whenever there's everybody goes in the same direction. So it turns out, just a quick summary, right? It turns out that this entorhinal cortex also has world-centered receptor fields or place cell type fields, but in a quite different way from the hippocampus. Now, what is that? What's the difference here? And it's pretty cool. So in 2005, this married couple called the Mosers, they're May Britt and Edvard, they're Norwegian. They were looking at entorhinal cortex in a rat navigating, and they found something very similar to the place cells, like I already said. But they found these neurons that have place fields that are repetitive. See that? Like a repetitive pattern, where again, the red dots are firing of this entorhinal cortex neuron, and the black lines are where the animal is in some enclosure. Oh, that's what I did. So it's strange, right? Now you have something like a place cell, but it doesn't report a single position. So that's freaky, right? So how does the brain know where the animal is? Based on a pattern like this, it seems like you've lost a lot of information just now by having this repetitive pattern. So how does the brain use this? What is the sort of algorithm that this is implementing? Well, it turns out that if you look at a whole population of these neurons and you sort of look very specifically, what you see is that they are implementing really like a grid in world-centered space. So there's this grid-like representation, a hexagonal grid that they report locations of. And if you look at different entorhinal cortex grid cells, they differ. They differ in ways that are different from how place cells differ, because the place cells just have different place fields. But different entorhinal cortex cells, they have different frequencies of a repetition. So they have a denser, more densely sampled or more coarsely sampled pattern in this real world space. And they will be shifted relative to one another. So they'll have different phases relative to one another. But they will all have, so this is, like if you're looking at entorhinal cortex cells that have the same frequency, they will differ in terms of phases. But they will all have the same angle. So sorry, I just skipped over this. They will all have the same angle. So if you look at this cell, it has an angle like this. A neighboring cell will have exactly the same angle. So it seems as if something is up with that angle. Now, what is this relation between place cells and grid cells? It's really sort of baffling, right? You have this sort of input-output stage. It's like what gives the hippocampus its information and also translates back. And so how can you construct? So that's really the idea, right? If the entorhinal cortex is really the gatekeeper to the hippocampus, you need to be able to construct place cells from grid cells. So how do you do that? And so this is not something that we necessarily know for a fact, but this is what's thought. So this is the sort of main model that people have for how this sort of function works. Is that if you have a single bank of grid cells, you can reweight those grid cells, so like sum them up in a different way, and then produce different place fields. So what does that look like? Well, let's say you have a bank of grid cells. They differ in terms of their frequency, right? So coarse to fine. And you're going to map those by summing them together with some set of weights, and then saying like I do a winner take all sort of thing, and then you get a place field. Yeah, that's the idea that we can construct the place cell from a set of grid cells. Okay. So that's with weights x1, x2, x3, x4, and x5, you get this output. Yeah. What I'm now going to do is I'm going to put a different weighting vector in front. So I now have y1, y2, y3, y4. Right. I'm just going to multiply those with the same set of grid cells. Right. So the grid cells remain constant, but I can change the weightings that they have relative to one another. And then by changing these weights, I can create a pattern like this. Again, take a winner take all type thing, and then I just shifted that place cell over there. So by having fixed grid cells, but reweighting their inputs towards hippocampus, I can flexibly change the positions of place cells. So what that means is, if the rest of the brain knows X and Y and these place cells, it can also translate back again. So this is the way that we think grid cells can encode specific locations in the outside world space. Is that clear? That's a sort of general mechanism. Cool. What did I say there? Oh yeah. So yeah. Just all you need to do, tilt your head. Yeah. So you can reweight these specific grid cells, right? Just make sure that you have flexible connections towards hippocampus, and that allows you to remap, right? So this is changing the weights amounts to remapping the place field. Clear? So any environment, you just implement a different set of weights, and boom, presto. That's the idea. Cool. So what we've sort of been talking about up until now. Well, first, like last week, we talked about these sensory bases, right? And vision in terms of sound, frequency, and then you can go even to more abstract spaces like even semantic spaces. And I want to just briefly spend a little slide on how do we know where we are and how do we know where things are in the world? And how does our brain, what are the different strategies that our brains can use to know location, either in sensory coordinates or in real world coordinates? For those of you that are really into linear algebra, a bit of this will sound a bit sort of intuitive, but I think that's the best way of trying to describe these things. So let's say, on the one hand, we have these receptive fields in sensory systems, which you can see as a Gaussian basis, right? So you have a way of sampling a space by means of these Gaussian blobs, right? And those parts of the space that you find more important, you're going to have more finely sampled, and every neuron there or every unit has a smaller receptor field. You also have more detailed information for every neuron, and that you might call a Gaussian basis, right? So that would be, you have sensory coordinates X and Y, you would tile that space like so, right? And this is in the case that it's a uniform sample. So if you would be able to linearly combine these with some weight matrix X, you can encode location in sensory coordinates, right? So that's like the general sort of processing strategy that we use in vision, sensory coordinates, frequency space, and audition, that sort of thing, right? So our brains want to know what the pitch of sound is, for example. You code that in the sensory coordinates. Yeah. But what we've just seen is quite a different way of encoding space, especially when it comes to these grid cells. And what we think happens in these cases is that what the brain is doing is it's just switching from a different basis, right? And it's not doing things in a sort of Euclidean Gaussian basis, but in a Fourier basis, where things are described in terms of frequencies, sines and cosines at different frequencies. And so what you then have is, in world coordinates, you have this scheme where you have things varying in terms of phase and varying in terms of frequency, right? So not X and Y, but phase and frequency. And if you can take a specific linear combination with different weights and then encode location in world coordinates. Yeah. And you could, in principle, also encode sensory coordinates with the Fourier basis, but that's not the general thing that the brain evidently does. Right? It's chosen to use this Fourier basis for world coordinates mostly, and this sort of Euclidean Gaussian basis for sensory computations. Right? But again, this gives us the problem of how does it go back and forth, right? That's still an unsolved question, but this is a way of thinking about how the brain is approaching this in terms of like the mathematical framework that it uses for encoding locations.
Is it maybe because also auditory is also used for coding with space? Okay. Then that's not directly translatable to visual coordinates.
Oh no, so yeah, well, this is really interesting. So auditory location actually is many different sources of information at the same time. So there's your brain has a model of the shape and attenuation profile of its own head. So if I'm holding my head like this, my brain knows that if you're talking, the sound here will arrive earlier, like with this like slight difference in location, like in distance. This ear will hear you earlier, and it will hear you harder, louder. And so these are two separate sources of information that your brain then uses to distill where things are on this axis. And then your brain uses the frequency attenuation profile of the ear to do height. So that's what allows the brain to know what's where. Yeah. And the problem is that's stuck to your head. Rotate your head, that all changes. Right? So that's not world coordinates. That's some intermediate level between the two, right? So in terms of vision, that's stuck to your eyes, right? So whenever you move your eyes, that's problematic. But then when you move your head and your eyes, you have two levels of total distortion, right? So it's thought that the audition and vision are brought together at the level of craniotopic representation. So that's when the vision has already accounted for eye movements, but their still vision is still sort of in head-centered coordinates. And that's when they sort of come together. But you're still not in world coordinates. Does that sort of answer your question?
Maybe, yeah. I'm also just wondering, why, yeah, you were asking yourself, why is it doing it? So, yeah.
Ah, yes.
Why is it doing a Fourier transform or Fourier basis, using Euclidean?
Ah, yeah.
It's more of an evolutionary question, I guess.
Yeah, I think for the given problem space, I'll talk a bit about that, by the way. So this is more versatile. This is more general, and you can repurpose it easier. And I'll show you an example of how you would do that. But this is just to give you an intuition, and I'll show you how this is more versatile a little bit. This is all nice and good. You can record from single electron with single electrodes, single neurons in animals. You can't really in humans, and so you need to come up with an fMRI experiment. And here it starts to become really interesting and important for this experiment, at least for you to understand this experiment. That all these grid cells have more or less the same angle. Yeah. So they have the same angle. They differ in terms of phase and they differ in terms of frequency, but they do have the same angle. Okay, so if we want to do an experiment finding this grid-like coding in humans, how do we do this? And in 2010, this dude called Christian Beller did this experiment that was that's a really intriguing experiment. So the thing is that he first found in animals that the grid cells all have identical orientations. The Mosers found this in 2004, 2005, this is a couple of years later. And then the idea was to perform a sort of VR experiment in fMRI. So you just let people navigate this sort of world. So there's a circular arena. You're in this world, right? So you navigate, you have like a set of buttons that you walk through this world. With different speeds, and you can just run around in this world, and your knowledge of where you are is because of there being mountains outside of the arena, so you know what direction you're headed because certain mountains are to your left, to your right, that's it. So what you have as an experiment, people were supposed to find things in this space that would only become visible if they were really close, so they'd be running around foraging for specific things. And so in their experiment, what you have is you have people in the scanner, they're running around all the time, running back and forth, back and forth. And the question is, okay, how can you use the data that come from this experiment to distill whether there is grid-like coding in humans? Well, what they did is they had this really interesting idea, which is, okay, let's say you have this grid in your head, right? And if you're running along the grid, right? You're going to go like bump, 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 right? And then if you're doing that in this direction, you're going to have a lot of activations happening of these grid cells, right? They're going to activate a lot. If you're running along the angle of the grid. But if you're just running just a bit off, right? You're going to go like, okay, I'm hitting a cell here, and then the next cell I hit is there, which is like almost 4 times the distance, right? So you have 4 times fewer moments of activation cells. So, and this is when you're running, right? So this is depending on the vector of movement, really. So what we expect in the fMRI signal then is if your foraging is in line with some angle, that's the grid angle, you have very high activations in these regions in the brain that perform this grid-like coding. But if you're just off it, you don't expect this high activation. So you can actually divide the circle up into, pi pieces of high activation and pi pieces of low activation. And because this is always a hexagonal grid, that's something that we know from the grid cells, this is always 60 degrees, modulo 60 degrees. And so what you can then do is you can try to find whether, as a function of the running direction that people have in the arena, whether you find a periodic signal at 60 degrees of this direction. So you have this is the red in this pie chart, and this is the gray in this pie chart. And so the prediction really is, okay, we're going to find this activation that's periodic, modulo 60 degrees, as a function of direction of movement in the arena. I've always thought this is a very elegant experimental design, right? So just thinking deeply about what it means when, if you were to have this organization, and then really devise an experiment that allows you to test this. So what they found was indeed that in human entorhinal cortex, which is in humans right here in the middle of the brain, they found these a cluster of voxels in entorhinal cortex that showed exactly this periodicity, like 60 degrees sampling. It was strongest when people moved fastest, which is also what you predict, right? You get more humps that you traverse. And they also tested different periodicity moduli, in other words, like you could do it for 90 degrees or for 45. And in other papers, they've done like more finely sampled. And it really is like a 6-fold symmetry, like 60-degree modulo. Yeah, and this also correlated with how well people remembered where they had been in the arena. And so post-hoc memory test. So what they did is they, based on the neural firing patterns, these grid cell signatures, they devised an experiment to to test this. They thought of a certain signature that the signal should carry, and then found this Nature paper. Yeah, beautiful paper. Yeah. So yeah, this is really how, like, I think the Nobel Prize was in 2015 or something. So this is really what brought this field to, like people can do it in humans, or found it in animals. New way of thinking about how the brain encodes space. So Nobel Prize in 2015. The Nobel Prize was awarded to the Mosers and to O'Keefe and one other person. By now it's a whole field. So this is all nice and good. Right, we found something in mice and rats, and apparently we can also find it using it for mining humans. But humans, we are so special. Don't you agree? We can do so much more, right? We think of things that a mouse could never think of. Let alone, well, not, and a rat either. Rats are maybe a bit smarter than mice. I don't know if there's a program that I can think. Yeah.
Make music,
What?
make music, for example,
Yeah, we can do that.
because music in some way is sort of abstract, it's spatial, it's
Oh, yeah, that's nice. Yeah. Oh, that's a nice idea. Okay, I'm going to show you this experiment that's on this slide. And then you should start musing about how you would do an experiment with music. Okay. Yeah. Because yeah, we have so many different ways in which we can think abstractly, right? About what is in the world. We can think about things that are not in the world. We are the kings of abstract information processing, right? Right? Very good of us. And so this paper that I'm going to show you now is from, I think, 2019 or something like that. So even more recent. And it's just kind of funny. And I think it's, again, an idea of an example of if you have a model, or at least a very clear sort of idea of what type of experiment you should do, then you can do that experiment and really prove a very insightful point about how the brain works. Right? I think you just saw an example, right? You need to do like a sort of model-based reasoning of the geometry of the grid pattern, and then see how that relates to fMRI signals that you just saw. And this one is just a bit more playful, and it's a lot of fun. Talk too much on an empty slide now, so I'll just start now. Yeah. So this is a paper that was 2016. Really that old? Anyway, yeah, it's a paper published in Science. I'm going to put it on the. I'm going to put a review that also talks about this paper on Perusall. It's not going to be part of the exam materials, but which is fine. So they said, okay, we have this cognitive map, right, from the Tolman stuff. Yeah. But we can think in all these different dimensions that maybe also have this weird, like a spatial layout. And you know, there's this abstraction to world coordinates. Maybe the same type of coordinate system is used in the brain in arbitrary abstract dimensions. So the idea is really like, if this is a general coding principle, this grid-like coding, then maybe our brains use it for everything, right? And that's that versatility that I was just talking about, right? This Fourier basis, using it to encode information in any two-dimensional space. It's really good for that. It's very, and so what they hypothesize is that if you have people do some task that involves abstract conceptual representations, that you should also be able to find that 60-degree periodic type signal that I've just outlined for navigation. Yeah, and it's a nice idea, right? So like, okay, maybe this is general. Let's do an experiment. And they came up with this. So, yeah, hang on. They devised a two-dimensional, yeah, he's gonna be proud. They devised a two-dimensional space that would have to be as weird as possible, right? A two-dimensional space that you never encounter in everyday life. Their two-dimensional space is associated with is like the two dimensions are the length of the neck of a bird and the length of the legs of a bird. And so what they allowed people, what they had people learn is where these things associated with rewards were in this space spanned by the neck length and leg length of these birds. Yeah. So you're learning this. Imagine you're in this experiment, like you go, you get paid 15 euros an hour or something. And then the experimenter asks you this, like, okay, now you need to learn this. I'm going to show you different birds, and then you have to tell me which of these icons is closer to your present position in terms of neck and length space. Good luck, bye-bye. Like they're like, yeah, they have to learn this sort of really weird task. And that's the point of this experiment, right? To make it a totally weird sort of space. Yeah. Just to prove a point. And it has this nice playful thing to it too, which I think adds. Okay, so if you think about this space as just legs length, neck length, these different elements are all in different locations in this space. Yeah. And so you can actually learn the topography of the space. And so they taught people the topography of the space. Yeah. Like, so that you could actually quiz people like, we're putting you here. What element is closer to where you are now? And then people would be able to say, it's a sled. And then, okay, so you know the two-dimensional space. You just learned the layouts of the two-dimensional space. That also means that you can show people a specific bird and then increase or decrease the length of its neck, right? Or increase or decrease the length of its legs, and that will give you a vector in this two-dimensional space, right? Because you're changing one or both of these two variables. So you can create like a path in this space by changing the layout of the bird. It's kind of weird, but you know, it translates, right? It's two-dimensional space. It's just, you haven't ever seen it. You just learned it 2 hours before you went into the scanner. And now you're going to do this experiment where I'm going to show you what the experiment was. So again, we're trying to find this, right? This is the goal. We're trying to find the 60-degree periodicity. And so these vectors that you can make by stretching birds in specific ways, they represent vectors in this space, and they have a direction. So if this direction aligns with that pie chart, then you know. Yeah, this should be a movie. Jesus. I'm pretty sure that it is. Oh, it is playing. Oh yeah, there you are. Yes. So there's a vector there. Right? We only changed the neck. And so the point here is that people learned this stuff about these birds, right? But they were never exposed to this being an actual two-dimensional space. This is implicit in the experiment, right? And, I think you can imagine doing this experiment and not necessarily being aware of this sort of two-dimensional layout, right? So the idea is then you have the entire experiment just consists of people looking at birds, changing, stretching, squeezing. And then they're adding a lot of birds here, but you get the idea, right? So all of these vectors are pointing in some direction. You can align them all with this expected periodicity. And you definitely would expect that on certain trials where you show this bird, for example, you get a high response, whereas something 30 degrees next to it, you get a low response. So they did this analysis and then found exactly entorhinal cortex and a few other regions in what we call the default mode network that also are very strongly connected to entorhinal cortex to have this sort of hexagonal coding. This paper was published in Science. It's a nice, playful thing yet again. So even though the sort of model-based part here is a bit distant, right? If there's this Fourier basis with grid-like coding thing, the experimental manipulation is weird, and the expected result in terms of the specific data hypothesis, very, very simple. I do think this is a very nice example of how we can use a model of how neurons function to learn about how the brain works, especially in these types of interesting cognitive domains, right? There's a certain vagueness to interesting cognitive experiments in many other fields that just seems to not be here with this specific experiment. I hope you agree. So, the question remains, how do we get from this sort of Euclidean Gaussian basis to this Fourier basis where we encode things in world coordinates? We don't really know how the brain does this because, and you saw it from your question, right? There are so many layers, right? You want to have things in headspace, because of sound is in headspace, but vision isn't. It's in eye space. So you have to, but then you can move your head again, and then whoa, everything is, then you need vestibular information to know which direction your head is pointing. There's so many layers. We don't really know how that works, but there are a few indications. I think the main idea here is that you need a lot of different systems to be able to create this abstraction in a truly abstract way, because you can use many different sources of information to create this abstraction, right? So not just in vision, also audition, but also like navigation. So here's a few examples, right? So we know that this entorhinal cortex is connected to a whole bunch of visual and association areas throughout the brain. I just showed you in this grid-like coding experiment that you saw these different regions also having this hexagonal grid-like coding. So we don't go from sensory coordinates to these abstract coordinates like directly. There's actually like 30, 40, 50% of the brain devoted to creating this abstraction to go to the level of the abstract coding of the hippocampus, right? So I just want to highlight a few of these different sources of information that this whole distributed network leading into the hippocampal entorhinal cortex complex, as people call it, what cues these are. So you have things like self-motion, right? You can think of that as a visual vestibular type of signal, where you just integrate like, oh, now I moved 3 meters to the left sort of thing, right? And I can feel myself stopping and that sort of thing. Very basic, but important cue for this type of information. Now, of course, we also have a lot of experience interpreting the scenes that we're confronted with. Yeah, and that's not just, am I standing in front of a church or a school building, right? Or is this my parents' living room, or am I in student housing or whatever, right? It's much more active. Right? We also saw one of those components like navigation and action are very important. When you see a scene, you don't just have a representation of its depth or what it is, right? Here you see a house and a car. It's not just labeled house and car. It also has surface inclinations, right? And so you have a 3D model of what that structure in the world is. You then move, you get a better idea of what that structure is in the world. But also important is that whenever you see a scene, you know exactly where to walk, right? So there is navigation inherent in seeing a visual scene, right? And we know that these regions that in the brain that do scene processing like this, they're actually in these very high level visual regions that you might call like a first or second association area. So you definitely, we do have an idea of, you know, that the different steps that might be necessary to go to this sort of abstract reasoning. It's just that the type of processing that happens in a brain that does this is extremely complex, right? It's quite different from an oriented receptive field in V1. It becomes a very complicated receptive field that likes, I don't know, not too bendy routes on wooden floors, or I don't know what matters, right? The idea is that we still need to find out, and we just don't know how the brain encodes this specific type of information. But we know what types of information are probably necessary. Now, if you go to this SFM, this conference that I went to, you see a lot of people going into this type of research. And one of the reasons is that we want to understand how this grid-like coding works. We also want to understand how to build it into a neural network. And there's a lot of work going into that. And there's a recent paper, relatively recent, that is, you know, sort of cobbled together a bunch of AI methodologies, neural network type methodologies, to create a neural network where they could look at the units in the network and then show that these had this sort of hexagonal coding type scheme there. So it is possible to recreate this. So this Nature paper was actually a collaboration between people at DeepMind and UCL. So the people doing this work were actually a team of neuroscientists and a team of sort of AI computer scientists. And I just want to show you briefly a quick movie about what they did. This is a very promotional video, but I think it's interesting. So I'm going to take this out, and I think I'll just play it, I guess. Wait.
Grid cells are a type of brain cell. And first of all, they were discovered in rodents, but now we know they exist in mammals in general. They seem to be very important for sort of measuring out space. A good analogy is that they're like the grid lines on a map, except on a map, we've used square grid lines and the brain's using hexagonal grid lines, and you have small scale grid lines and larger scale grid lines. Originally, people used to think they were very important for something we call path integration, which is basically updating where you think you are based on how you're moving. So I know where I am right now, and if I move 10 meters, then I end up somewhere over there. But actually, we're moving beyond that now. While we still think path integration is important, focusing on the fact that we believe they function like a map, they sort of relate one place to another. They also seem to be very important for planning routes. So if you want to know the shortest route between here and somewhere else that you visited, then the grid lines will provide you with something called vector-based navigation and allow you to calculate the direct line distance. With the recent paper, we were trying to do several things. So first of all, we were trying to test this idea of vector-based navigation. So are grid cells really the substrate, the machinery that allows you to sort of calculate these direct line routes between places? But equally, we're trying to do something a little bit more than that. We were trying to see if you could take what we know about how the mammalian brain works and incorporate it into artificial agents to improve the way that they work, in this case, the way they navigate. First of all, we were able to show that if we train an agent or a network to path integrate, then we found that actually it developed grid cells. So this is a remarkable convergence form in that they look like grid cells in the mammalian brain. They have the same hexagonal pattern with sort of tightly tessellated fields. And then we were able to take that network and transfer it into a larger network that was now controlling an agent navigating around. And that agent was then able to navigate flexibly in demanding virtual environments. And so, for example, one of the tests that we wanted to see whether it could do is whether it could take shortcuts. We had environments that would change while the agent was in there. So it might find one long route to the goal, but then a new door would open up presenting a shortcut. And so the agent with grid cells could utilize that shortcut and go straight through to the goal. So this is important for several reasons. We've confirmed that grid cells are important for vector-based navigations. We've shown that we can use an artificial network to conduct neuroscience experiments, but also we've shown that incorporating what we know about how the brain solves a problem into an artificial network can provide that network with abilities that it wouldn't have otherwise have had.
We can put money, I think, on the video. But you get the point, right? The idea is that now that we have these concepts from neuroscience, it's a beautiful idea to see again, like we've seen this with convolutional neural networks, right? Beautiful idea to see how these neuroscience findings are actually also going to play a role in navigation network. That's it for today. Yeah, I'll see you on Friday. It's a computer practical. It's going to be, a bit of both. But yeah, I'll also be there this time. Yeah. And so this evening, I'll send you which group you belong to. Right? It's nice to have a feeling of belonging halfway through the course. And you'll be able to start working on your podcast presentations. Yeah. Excellent. Yeah. Oh, the second one. Oh, you already finished it first. I'll put the second online. Yeah, that makes sense. I'll put it also online tonight. Yeah, so I'm going to like that final sort of podcast presentation lecture thing. I'm just gonna set up a timing thing, but if you have a problem, maybe it could be somewhere else or something. Yeah, so I'm still debating because I went to this conference, right? And I saw all the cool things that I want to picture. I'm still debating whether I'm going to add a lecture of my own and maybe got it rush to podcast presentations or the other way. Yeah, I'm gonna be. Yeah, right. It's like state of the art. Yeah. We'll see. Thinking very hard about what I should be doing.
One question
Yeah.
regarding the reset, because I'm not sure.
The reset, which the remapping thing, or
Not the
the reset.
more
Oh, yeah.
formally the
Hmm.
reset of the exam, because I couldn't find date yet.
Uh-huh.
And the 23rd, I'm not here anymore. That's why I was wondering whether I should maybe plan with the reset directly.
Yeah, that makes sense, because I am feeling a bit, let me stop and the recording.